Thank you very much, Matthew, for this very kind introduction. I will share my screen. Okay, wonderful. You should be able to see it now. I'm just going to start the presentation. 2 seconds. Voila. Great. Yeah, it's my great pleasure to speak here today on regulatory T cell stability. It's one of the focuses in my laboratory. Just going to choose laser pointer. Voila. Maybe first starting with a few words on the relevance of regulatory T cells, Treg stability, and also Treg plasticity. And these two are interrelated. Regulatory T cells, I assume most of you as immunologists will be aware, are absolutely indispensable for immune homeostasis. If they malfunction or if they are lacking, that will result in autoimmune diseases of different kinds or even the lethal IPEX syndrome. And the hallmark transcription factor for these T cells is FOXP3. That means FOXP3 is required for the development of regulatory T cells, but also beyond that for the homeostasis, for the stability, and for the function. Most of the regulatory T cells develop in the thymus, where the progenitor cells receive a moderate strength agonistic TCR signaling, excuse me, and then initiate the expression of FOXP3. And these thymic Treg, they still dwell in the thymus than before they actually egress, which gives them a little bit of an advantage over in terms of stability over peripherally induced Treg, that is Treg that convert in the periphery from naive or conventional CD4 T cells. Treg are then generally assumed to be a stable cell population, and that is mostly based on their epigenetic profile. Regulatory T cells need to possess a particular epigenetic landscape to allow this stability. A certain demethylation profile has to be there, and I'll get to these details during my presentation. However, besides that functional stability of the immunosuppressive functions, Treg also possess functional plasticity. That means Treg can specialize into certain T helper-like Treg. So then they do not only express the transcription factor FOXP3, but also the transcription factor that needs to be induced for the specialization of the respective T helper cell line that they want to co-localize with and want to suppress. This functional plasticity is very, very relevant. If it's missing, it results in disease. If Th1-like Treg are missing, you will have Th1 pathologies. Different Th-like Treg cannot compensate for each other, so they are very specific functions. But this plasticity, because these Treg also express a T helper transcription factor, the hallmark transcription factor of the respective T helper subset, that also unleashes a certain transcriptional landscape of that is T helper-like, that is normally suppressed in Treg. So now these Treg have to balance the pro-tolerogenic and the pro-inflammatory landscape that are now coexisting in these cells. And if this balance skips and is imbalanced, then the Treg will lose their stability and may irreversibly become what's called an X-Treg. So then they will lose FOXP3 expression and will become highly pro-inflammatory. And they will express then, or they will acquire effector T cell characteristics, such as pro-inflammatory cytokine expression. Now, this is all very relevant. We need to understand Treg stability. We need to understand the impact of Treg plasticity, because Treg are more and more becoming an interesting target in cell-based therapeutics. The reduced suppressive function of Treg, or even the loss of Treg fate memory and the giving rise to X-Treg, can be caused by a number of different threats that a Treg can be exposed to. That is, it can be defects in immunosuppressive mechanisms. It can be perturbed homeostasis or hampered Treg development. But most importantly, it all comes down to epigenetic instability. If the epigenetic stability of Treg is not intact, then these cells won't be able to maintain their pro-tolerogenic transcriptional profile, epigenetic profile, and their function. And there's a lot of mechanisms that, on the one hand, assure Treg stability, and on the other hand, actually are a threat. So I'd like to just briefly give an overview of some of them, such as signaling pathways. Yeah, there are signaling pathways that are absolutely important for Treg stability, for Treg homeostasis. Yeah, TCR signaling, co-stimulation, IL-2, pSTAT5 signaling, for instance. And then there are signaling pathways like pro-inflammatory cytokine signaling pathways via STAT3, STAT1, STAT6 that are a threat to Treg stability. Very important. This all always comes down to the epigenetics. A threat to the epigenetic landscape of Treg are chromatin remodeling or organizing proteins such as SATB1 and methylases like DNMT1. On the other hand, there are mechanisms to promote Treg stability. Yeah, demethylation by TET enzymes. Then, of course, the hypomethylation of FOXP3 itself, the CNS2 region of FOXP3 itself, of the Treg signature genes, CTLA4, EOS, Helios, GITAR, etc. And EZH2, maybe we get to that later as well. Transcription factors, of course. Transcription factors that are related to pro-inflammatory transcriptional circuits are a threat to stability. I mentioned STAT3 already. Similarly to STAT1 or STAT6, they have to be very tightly regulated in Treg, otherwise they will hamper FOXP3 expression and initiate T helper-like gene expression. And then there is a whole range of transcription factors that are promoting Treg stability, such as BAC2. BAC2 is a transcription factor that actually restrains others like IRF4 or GATA3. STAT5, of course, downstream of IL-2, SMAD3, downstream of TGF-beta, etc. Other intrinsic pathways, P10, if Treg lack P10, they immediately lose stability and become X-Treg, so P10 is really key, but the PI3 kinase and AKT pathways downstream of a number of signaling receiving receptors, TCR is just one of them, are a potential threat. PI3 kinase AKT is also downstream of glycolysis. Unconstrained glycolysis is actually not very much in favor of Treg stability. They rather prefer OXPHOS or also for peripheral Treg conversion, metabolic components such as retinoic acid or SCFA, short chain fatty acids. And last but not least, post-translational modifications. For instance, FOXP3 protein itself is heavily post-translationally modified. For instance, TIP60 or P300, they acetylate FOXP3 to increase its stability, its protein stability, while sirtuin-1 actually does the exact opposite, it deacylates, and then thereby, of course, presents a threat to stability. So what I'm trying to say with this is that Treg stability is very neatly regulated and is a very complex matter, actually. But what I'd like to highlight is that most important to Treg stability, and I think the literature agrees on that, is the installation and maintenance of the epigenetic landscape, the Treg-specific epigenetic landscape. That means that Treg-specific genes, relevant genes for immunosuppressive mechanisms of Treg are hypomethylated, and that pro-inflammatory genes or gene circuits of helper, T helper subsets are suppressed. And all this, if looking at thymic Treg, where we assume that most of the Treg are thymus derived, starts in the thymic progenitor that can actually still give rise to conventional T cells or conventional CD4s and Treg. But then the activation of a super enhancer region by SATP1 leads to a genomic reorganization. And this genomic reorganization somewhat opens up Treg-specific loci and allows for the recruitment. The next slide. Sorry for the delay. Voila, there we are. Allows for the recruitment of TET enzymes. Now, initially, there can already be FOXP3 expression, although most of the loci will still be heavily methylated. And the hypomethylation of Treg-specific loci and the already present expression of FOXP3 are not necessarily going hand in hand, they're more or less going parallel, and then it merges together in the process of gradual demethylation of the Treg-specific demethylated regions in different Treg-specific genes, all coordinated by the TET enzymes. And so then basically these are the Treg that are ready to egress, but they actually still dwell in the thymus, as I mentioned, for a couple of days, so that this landscape can be fully installed. This is one of the reasons why T-Treg are thought to be more stable than P-Treg, because when P-Treg try and do the same thing in the periphery, they may already actually encounter extrinsic destabilizing signals, while they're maybe still, much further down that process here. And the hypomethylation state is not yet reached. In the periphery, the Treg hypermethylated profile then needs to be maintained. And there's a number of factors actually doing that. The TET enzymes certainly are very important here, but also histone acetyltransferases. They acetylate different lysine residues and thereby increase accessibility of the loci and gene expression of Treg signature genes. UHFR1 together with DNMT1. That's an interesting one because per se, this is a methylase. And on Treg signature genes, well, you don't want them to be methylated again. They have just been demethylated and you need them to be methylated for stability. So the action of DNMT1 on the Treg loci is detrimental for Treg stability. However, it is still expressed in Treg because actually it methylates the T helper loci, yeah, and represses those because the epigenetic profile in Treg is not only about the hypomethylation of Treg genes, it's also about the methylation and control thereof of the non-Treg genes, the pro-inflammatory genes. And this is what DNMT1 does. So if DNMT1 is too little expressed in Treg, then the pro-inflammatory genes will start to be expressed, and that's detrimental. If it's too highly expressed, then it will also start to act on the Treg signature genes, and that is also detrimental. So DNMT1 is important, but also needs to be very carefully regulated. CORST is a repressor complex and is also involved in the repression of T helper genes. And with this, the Treg should be stable and should remain stable. But in fact, we find X-Treg in normal mice. So what's the nature of this Treg instability? So 3 models have been proposed for that. The stochastic model, the heterogeneity model, and the transient flexibility model. Is there one truth for all contexts? Surely not. Yeah, and towards the conclusion, I'd like to bring these models up again. There is arguments for every single one of them. So that's why likely we need a more holistic model or more context-dependent look at Treg stability. The stochastic model suggests that basically any Treg can lose FOXP3 expression and the immunosuppressive function if it's found under destabilizing conditions. The heterogeneity model instead suggests that Treg are a heterogeneous population, and that the population, well, it's what we all know, but that is also this population contains stable and unstable Treg clones. They are not very well defined, certainly not with surface markers, and that stable Treg will also basically remain stable, while unstable Treg can lose FOXP3 expression. And like this is basically a black and white picture looking at Treg. The transient flexibility model suggests that during an immune response, just show all of it at once, the effector T cells get activated, they mount the immune response, and towards the resolution, some of them may actually become induced pTreg, but mostly we'll be left with a few central memory T cells. The transient flexibility model for Treg actually suggests that, let's say, very locally, FOXP3 Treg during the mounting of the immune response can lose FOXP3 expression, but keep the fate memory. Yeah, that also would imply that they keep the epigenetic profile. So they become X-Treg pro-inflammatory to support the mounting of the immune response, while later, towards the active and towards the resolution phase, these X-Treg would spontaneously revert back to Treg and allow the the control and the resolution of the response. Now, there's not many examples for this actually happening in the literature. In fact, I only found one, but I think it's very difficult to demonstrate. And also, this may happen very context dependent and very local. So X-Treg, well, if Treg are very stable, then we shouldn't find many X-Treg. But when we look into, when we analyze Treg reporter mice, fate mapper mice, where the FOXP3 locus is linked to a to expression of the Cre recombinase, and the Cre recombinase recombines a reporter locus, ROSA RFP. It removes a transcriptional stopper and puts the RFP in place. And what's happening in these mice is that all the Treg will express the YFP because it's part of the FOXP3 locus. And it will also express Cre, so they will all be YFP and RFP positive. But when they lose FOXP3 expression, because they become X-Treg, then they will only express RFP and will have lost YFP expression. And like this, you can identify X-Treg in these mice. This is how such a flow plot would then look. Yeah, you have the double positive Treg and you have the single positive, the RFP, only positive X-Treg. And actually, this makes quite a large proportion of cells. It is percent X-Treg of CD4, not of Treg, of total CD4. So it's quite substantial, about 4 to 6% of CD4s are actually X-Treg. Now that does not necessarily result in disease. These mice are perfectly fine and healthy. It's a normal wild-type mice besides having 2 transgenic alleles. So they're well controlled, but it also means that there is, even in normal healthy mice, quite some loss of Treg into X-Treg. And these X-Treg, as you would expect, display a very pro-inflammatory phenotype. The X-Treg here are in red, the Treg in blue. So you can appreciate that they express then high levels of IL-2. More than 50% of them actually express interferon gamma. A lot of them will also express TNF-alpha, and they have lost TGF-beta. So these are now pro-inflammatory cells. So where do these cells come from? Which Treg will preferentially give rise to X-Treg? So we've looked into this in great detail. I'd like to share some of the data with you. And then also in the end, discuss the impact that Treg plasticity can have on X-Treg development. So what we've done is we performed a T cell transfer model into lymphopenic mouse. These are REC1 knockout mice. So we purify Treg as well as naive T cells. The naive T cells, upon activation, we require those as a source for IL-2 for the Treg. These are co-transferred, congenic, they're co-transferred into the lymphopenic host. And then 5 weeks later, we'll find, not too much surprise, this is a very well-known model to induce X-Treg. We'll find remaining Tregs and we'll find X-Treg. So we wanted to understand whether the stochastic model actually holds. So if X-Treg formation is stochastic, then we should be able to retransplant or retransfer these Treg with new naive T cells again into a REC knockout mice. And then some weeks later, we should find X-Treg again. In that case, I think it's fair to conclude that these Treg have not been all stable and that they have, some of them will have lost FOXP3 expression, even though they survived perfectly fine the first primary transfer. Or we will not find X-Treg. And then this is a very stable population purged of the unstable subset. So upon first transfer, upon primary transfer, you can appreciate here that there is quite a substantial ex-Treg population, and this example is 20%. While upon secondary transfer, voila, upon secondary transfer, this population is basically gone. Yeah, there's a few cells in this FOXP3 negative gate, but it's not actually even forming a population. And what we see is that the decrease in the ability of giving rise to ex-Treg, and we've performed this experiment 7 times, is about 85%. So this population, once purged of the unstable clones, is highly, highly stable. So what are these unstable cells that give rise to the ex-Treg? A number of different subsets have been proposed to give rise to ex-Treg. In the literature, you'd find that CD25 negative Treg are prone to give rise to ex-Treg. That would make sense. CD25 is part of the trimeric high affinity IL-2 receptor, highly expressed on Treg normally, and the IL-2 p-STAT5 signaling is supporting FOXP3 expression. p-STAT5 directly binds to the FOXP3 promoter. So 25 negative Treg, yes, they might be less stable. How about neuropilin negative Treg or uncommitted p-Treg? So neuropilin 1 has been suggested as a marker to enrich for p-Treg, and that is certainly true, I would say, in organs such as the gut. In other peripheral organs, I think this is a more controversial marker for peripherally derived Treg. I will later also show you some data that support this controversy on this marker. But beyond this being used as a marker, neuropilin 1 signaling also, downstream signaling also supports Treg stability. So there could be 2 ways of looking at this. Yeah, neuropilin 1 negative cells miss a stability signaling, or they are enriched in peripherally derived Treg, which are not yet entirely epigenetically committed. Okay, there's another option. Thymic Treg have also been suggested to give rise to ex-Treg. And we've looked with our model at a couple of these options. In the way we use our model now is basically we just use congenic naive T cells, and then we co-transfer to, again, congenically marked Treg subsets, subset A, subset B, that can be 25 positive versus 25 negative, and we can distinguish all 3 transferred populations by the congenic expression of CD45.1 versus 2. So first, maybe a look at CD25 negative versus positive Treg here on the left. And as you can immediately see, transferring 25 positive or transferring 25 negative Treg, neither population has a preference to give rise to ex-Treg over the other. Both give rise to the same amount of ex-Treg. And interestingly, 25 expression is just like many other surface marker expression, it's not static. So what we've seen is also that within the remaining Treg that have been transferred as 25 positive, we then find 25 negatives, or that have been transferred as 25 negative, we would then find then 25 positives. Yeah. So I would say that strongly argues against 25 negative Treg being a main source or a preferential source for ex-Treg. How about neuropilin? Neuropilin, now that if you follow the mainstream literature, we would say here that these cells are strongly enriched in peripheral derived Treg. And yes, the neuropilin 1 negative Treg strongly enrich for ex-Treg development. These cells are just much more unstable than the neuropilin 1 positive Treg. So then the question is it really as simple as P-Treg are unstable, T-Treg are stable? If I'm posing the question like this, then you can already imagine the answer. So to look a little bit into the details, the transcriptomic details that are happening during X-Treg development, during the loss of Treg fate, we've performed single-cell RNA-seq, or single-cell multiomics, I should say, where we've compared 4 populations. One is a normal ex vivo Treg population, just sorted from a normal mouse with the FOXP3 fate mapping or marker expression. So these cells contain everything: stable clones, unstable clones, not enriched for anything. And then we looked at X-Treg 5, 10, or 15 days after transfer into the reg knockout mice, so after the stability insult. And what we saw is that these the Treg and X-Treg populations, they cluster completely differently. So the Treg in red, they form 2 clusters, the naive ones and the activated ones. And closest to the naive ones, we actually find the cluster of early X-Treg. The early X-Treg do not at all cluster together with the late X-Treg, which are, day 10 or day 15. It doesn't make any difference anymore because they are so transcriptionally similar that they just intermingle in the last 3 clusters. So something is special to these ones. And what we've then done is a network analysis on your left, where we see that the Treg population splits up in 3 and forms this extra population that is now closest related to the day 5 X-Treg. And when we analyzed this in more detail, we saw that the day 5 X-Treg, the earliest X-Treg, they express very high levels of CD62L. That's actually protein level here even, yeah. So very high levels of CD62L. These cells are highly naive, and conversely, they express very low amounts of CD44. And it seems like that this population is somewhat enriched in this, what we call here the inter-Treg, this population that's closest related here. So we find an enrichment. And if we look for neuropilin-1, we also see that the inter-Treg enriched in these naive cells are also enriched for neuropilin-1 negative cells. So early X-Treg are very naive. So is it just sufficient to look at the activation status to identify unstable Treg? So we looked at that. We just sorted naive versus activated Treg, put them into the mice. 2 weeks later, nah. So naive versus activated Treg, yes, there is a trend, but just by looking at naive T cells, likely because the unstable Treg are such a small population in a large pool of naive Treg is insufficient to predict instability. So if we further dissect that by adding to the activation status also neuropilin-1 positivity versus negativity, even then there is not much of a difference in terms of being able to predict the stability or instability of Treg. So that brings me back to the question, is it now as simple as P-Treg versus T-Treg? If it can't be just activation status. So what we did is we used a mouse model, a transgenic mouse, in which a FOXP3 CRE transgene is only expressed by thymic Treg. Okay, if you cross this transgene to ROSA RFP, then only the thymic derived Treg will be GFP and RFP positive. we also, in addition, crossed in FOXP3 type 1.1 to be able to follow all Treg as a pool. Yeah, if we now look at the total of Treg, so we sorted just for FOXP3 type 1.1 and transferred this mix, then what we transfer is this top, yeah, Q1 and Q2. So we transfer the T-Treg, which are RFP positive, and we transfer the P-Treg, which are RFP negative. And after 2 weeks, we do get X-Treg, and we can perfectly identify which X-Treg arise from T-Treg, because they lack FOXP3, but they still have the RFP, and which X-Treg arise from peripherally derived Treg. They lack FOXP3, and in addition, they lack the RFP. And by just looking at the frequency of cells of X-Treg that we get from each population, it's quite clear T-Treg are much more stable than P-Treg. Yeah. Also here, you once more see that the peripherally derived Treg in red give preferentially rise to X-Treg compared to the thymic derived Treg. So, okay, the conclusion, and the same, that actually matches with what we've observed with the neuropilin-1 negative Treg. So from that, we could per se conclude most of the X-Treg in the periphery are of P-Treg origin, and there is no issue with autoreactive TCRs because P-Treg are not enriched for autoreactive TCRs. Is it that simple? Not really, and that is because there is just so many more thymic Treg compared to peripheral Treg. Yeah, so if you now look at the overall contribution to the X-Treg population, you'll find that the P-Treg and T-Treg almost contribute similarly. Yeah, the T-Treg almost seem to contribute more to more cells to the X-Treg subset. So there's a substantial contribution of the T-Treg to the X-Treg. So that's why we also wanted to have a look at the thymically derived Treg. But we haven't had another reason for that. We've had performed single-cell RNA-seq, and another finding that came from that is that an observation is that PD-LIM4 is highly enriched in the early X-Treg compartment and the day 5 X-Treg, while you would barely find it later on. Yeah. So PD-LIM4 is interesting because it's an adapter protein for S1P1, and S1P1 is required for the egress from secondary lymphoid organs, from lymph nodes, but also it is required for egress from the thymus. So that made us wonder whether maybe some of the RTE are giving rise to X-Treg in the periphery, and that some of the RTE have not yet fully locked in the epigenetic Treg signature. So we tested that by using mice in which you can identify the RTE in the periphery by GFP expression. In the interest of time, I'm not going to explain this in great detail here, but by looking at the thymic Treg, the Treg RTE phenotypically, we see it actually matches with what we've observed with single-cell RNA-seq and also with previous transfer experiments. The RTE are highly naive, and interestingly, half of the RTE will not even express neuropilin-1. So neuropilin-1 negative cells in the periphery are not necessarily peripherally derived Treg. Actually, this population also contains the RTE. And if they express neuropilin-1, then it's very low levels. So the neuropilin-1 negativity in the periphery means P-Treg and also RTE. So now are these RTE, I mean, they had some time in the thymus, maybe they are just perfectly stable. So we transferred them as well. And what we find is, in fact, they are less stable than older Treg that are already in the periphery for much longer. Yet they are more stable than the peripherally derived Treg, of which sometimes half of the Treg can give rise to X-Treg. So they have some stability issues still, besides being thymically derived Treg. So Treg instability is inherently problematic, as I mentioned, that thymic Treg are enriched in self-reactive TCRs. They become very pro-inflammatory, and if they have fate memory, like the transient flexibility model suggests, at least in some circumstances, then there's no issue because they can lose FOXP3 and they can regain it. But if they have no fate memory, then actually they can drive autoimmune diseases or contribute to the progression of inflammatory and autoinflammatory diseases. So we've tested this, and I'm just going to skip over this slide in the interest of time. We've tested this in vivo and in vitro, and we find little to no evidence for fate memory of X-Treg. So at least in our model, and that must be tested in other models as well and confirmed, the X-Treg seem to lose Treg fate memory quickly and for good. We've tested this in vitro and in vivo with the exact same results. I'm just going to skip over this. Voila. So this is all very relevant in the context of Treg functional plasticity, and I'm just going to spend the last couple of minutes on that. As I mentioned, Treg can, when they receive certain pro-inflammatory signaling, similar signaling that the respective T helper cell type would require for its polarization. Also, Treg can polarize into a TH-like Treg, which is what we call functional plasticity. So then these Treg, they co-opt this the lineage determining transcription factor of T helper cells, and are now double positive for FOXP3 and that transcription factor. This transcription factor expression is very important. T helper like Treg are very important. It is required for the cells to express the respective chemokine receptors to co-localize with their target cell, T helper cell type, to efficiently suppress. But if the programs, the pro-tolerogenic program driven by FOXP3 and the partial pro-inflammatory transcriptional profile driven by the respective T helper transcription factor, if these are unbalanced in favor of the latter, then this can result in Treg and X-Treg. And these X-Treg, they have been demonstrated to contribute and drive different inflammatory diseases, different pathologies for Th1-like Treg that can result in the downregulation of PTEN. I've mentioned that already. FOXO1/3 are not properly localized in the cells, and then that gives rise to type 1 autoinflammatory diseases. The Th2-like Treg are a little bit more complex because GATA3 and IRF4 are generally expressed by Treg, also required for a Treg phenotype. But once they are, and this expression is independent of IL4 and STAT6, but once they cross a certain threshold and are highly expressed, then they turn on the Th2 transcriptional pattern and become Th2-like Treg. And if this balance between FOXP3 and GATA3 is not correct, then this results in X-Treg. For Th17-like Treg, this is the only example that I honestly could find where a regain of immunosuppressive features has been described upon loss thereof. And TFRs are a great example for instability, because the Treg that then express BCL6 and migrate via CXCR5 into the germinal centers, they receive so much signaling, it's really overwhelming the cells, that between 20 and 30% of the TFR actually lose their Treg fate. Yeah. We don't really know whether this has any consequences, negative consequences, but it's quite impressive. So in conclusion, xTreg are there, they're detectable in healthy mice, and generally our data support a heterogeneity model of Treg instability, but it's one model or one experimental setup that we're using here. The plasticity-induced instability per se could also support the stochastic model, yeah, because the Treg plasticity is also not something static. A Th1-like Treg can become a normal Treg again, and then later circumstances become a different type of Th-like Treg. And only for Th17, like xTreg, it has been so far described that they can regain suppressive function that would support the transient flexibility model, but there's still a lot of work that needs to be done to either integrate all these data into one more holistic model or to dissect that further. So in conclusion, the unstable Treg are a complex subset that are enriched in naive pTreg and RTE. And it looks like once the Treg core signature is lost of these unstable Treg, that they very quickly cross the point of no return, and that the majority of them will actually lack Treg fate memory. So this could suggest that in terms of translation of all these kind of findings, that maybe we should not try and stabilize the Treg population in vitro before we transfer them into patients, because we don't really know whether this is working and whether all the Treg are then stable. We could actually do it the other way around, destabilize all the Treg, remove everything that has lost Treg fate, repurify, and then have a highly stable population that has been now purged of unstable Treg that could give rise to xTreg. And with that, I'd like to acknowledge members of my lab and collaborating labs in Belgium and UK. And maybe a last note, we are hiring. Good opportunity maybe to mention that. So we have a PhD position available starting in January on the role of RNA modifications in immune cells, in case you're interested yourself or you know somebody that may be interested. And with that, I'd like to thank you for your attention.
Thank you so much for that, Susan. That was so interesting. Really, really enjoyed that. First of all, on the PhD position aspect, we can also share this on our on the STEM Village Twitter page to let people know that you're looking. And if anyone has any questions for Susan, you can unmute yourself and ask, or you can write these in the chat box, whichever you prefer. Just to get the ball rolling, one thing that I was wondering was, when you were talking about the egress of the Tregs from the thymus, and that they may not have their full sort of epigenetic program in place at that point. I mean, this might be a bit of a tangent, but because so much of the epigenetic sort of modifying enzymes are influenced by, you know, for example, the TCA cycle, oxidative phosphorylation, things such as that. Is there any hint, or is it known whether the epigenome at that stage is influenced if you have underlying metabolic disease. So people who are sick, but also perhaps obese or diabetic. Is anything like that known?
Straight answer, we don't know.
Okay.
I don't know. I wouldn't be able to answer that. We have literally no data on that whatsoever. Maybe we should look into some obese mouse models for that.
Mm-hmm.
There is a number of models that one can look at.
Yeah, no, I'll have a look. Sorry if that was.
Well, I should have a look.
Yeah, it was just interesting because my background is sort of metabolism and epigenetics. So when someone talks about epigenetics, I'm like, oh, that sounds.
Yeah. It is really, really interesting. Keep your eyes open. We're just going to soon publish a nice review on Treg epigenetics and stability and Treg plasticity. So
nice. Nice. I'll have a look for that. So someone was saying, thank you very much for the report, for the talk. It was incredibly interesting. I think, and Juan, can you unmute yourself? Or
Hi, Susan. That was incredible. Thank you very much. I had a question regarding the. I'm just surprised. I know nothing about Treg, right? But I was just surprised to see how flexible and plastic they are. And that's fascinating. And I wondered if you have any evidence of markers that are retained epigenetically once these T cells kind of become ex-Tregs and then they gain the Tregs function again. Do you see kind of poise promoters or anything like that that will make the whole process easier, more efficient, or?
Yeah, so there's really only one publication that claims that that Th17 like Treg have turned into X-Treg and then have regained their immunosuppressive function again, have regained FOXP3.
Okay.
So I'm putting this always as like something very careful until I see it another couple of times maybe.
Yeah.
We have not seen this at all. I mean, our data argue for the exact opposite, but what we are working on is a model to roughly study what you're asking for. Yeah, so where we in a much more physiological setting, not a T cell transfer model. This is really, really, for the cells is very harsh. You put them in there and they just, they start to proliferate. They get all this cytokine storm and it's crazy for them. And then you get all these X-Treg. So what we would like to do is to do this in normal mice, lymphonormal mice, where we basically, in some sort of knockdown approach, knock down FOXP3 and some of the Treg. FOXP3 can be any gene. We're now choosing FOXP3 for now, and then see what's happening epigenetically over time, and how these epigenetic changes over time impact the transcriptome, but also their ability to return to the Treg lineage.
Yeah.
Okay. So and then we can actually see what is the epigenetic point of no return and how does that look transcriptomically, and maybe we can also then relate that to some proteomic markers where we can see like, okay, these Treg are about to lose it. Yeah. Exactly. This could be a target for stabilization. This could be a target for destabilization in tumor settings. Yeah. So this is what we're after next.
And I had another question, if I can, Matthew. Do we have?
Yeah, of course.
Again, this is me being completely naive. I have no idea about the field.
These are the worst, really.
So in terms of these X-Tregs, I mean, in my head, I'm thinking, well, what would be the sort of the advantage of becoming an X-Treg, physiologically speaking? Now, do they become like memory T cells? Do they become resident T cells? So is this X-Treg population sort of?
Yeah, I mean, for the X-Treg themselves, I'm not sure it has much of an advantage.
Yeah.
We also don't really know what it means for them homeostatically.
Yeah.
Yeah. Does it extend survival? Surely it will improve proliferation.
Yeah.
But I mean, overall, will they be longer lived than Treg? So we don't know that. What I find interesting is that there is apparently plenty of these X-Treg, and these are pro-inflammatory cells present in mice. Surely this will also be the case in human. Yeah, and yet per se they don't do anything. They're really well controlled. The problem really only starts when we have this local X-Treg development in the context of an ongoing inflammation. Then they will not by themselves necessarily, unless you have a specific maybe genetic predisposition or something, by themselves they may not cause, in these small amounts where this is happening for Treg plasticity, they will not cause an inflammatory disease. Of course, if you have a genetic predisposition, and all the Treg do that at the same time, well, then you're in trouble. But per se,
but if
the problem only starts once there is already an inflammation ongoing, and then they just go with that flow. There's insufficient, and then it becomes a vicious cycle. There's insufficient immunosuppression that again increases the extrinsic pressure on the Treg that are still there, and so forth. And like this, you create a loop that you really have to, break with immunosuppressants. And
of course, wow, that's so intriguing. Thank you very much.
Thank you.
One other question that I had, and I may have missed this when you were talking about it, because I didn't know previously that the Tregs acquire the transcription factor that's relevant to whichever T helper cell population that they're trying to control. Is there any plasticity in that, or is it a case of once they start, for example, expressing raw gamma T, that's it, they are now a raw gamma T FOXP3 positive T cell?
Good question. So my understanding is that there is plasticity, the extent of which I'm not sure we know.
Okay.
You hear me a lot saying that we don't know, but we just really don't know.
No, I mean, it's really interesting.
But I mean, they adopt this phenotype, and this phenotype, or this plasticity phenotype, and it's fed by cytokine signaling. I mean, with the loss of the cytokine signaling, and the balance being moved back towards FOXP3 pro-tolerogenic and away from, let's say, STAT3, STAT1, STAT6, then they will lose that they will lose the raw gamma T expression again, because that's all fed by STAT3. P-STAT3 gone, well, then they become normal Treg again. Per se, okay, then they're kind of already at location because they migrated there. Because the whole purpose of them becoming TH like Treg is to co-localize. Initially, it was thought that they have a specific suppressive mechanisms or something, and that's maybe true to a certain extent for maybe increased expression of CTLA4 for Th2 likes, or IL-10 production in the gut, in other barrier organs. But overall, their suppressive portfolio doesn't really change. So it's mostly about co-localization. But all this is regulated via cytokines. So once this breaks away, then basically they're still there. But per se, they don't have any further intention to migrate there. They can't follow the gradient any longer. And also the integrins to hold them there is also regulated via the cytokines. So per se, they could actually just enter the normal Treg cycle again. That's why I think there is probably a lot of plasticity. How many Tregs that are effectively doing? I mean, how often do you want to be sick enough to require Th1 like, Th2 likes? But I think per se it's possible.
Okay, well, that's really interesting. Cool. Well, if we don't have any other questions at the moment, I just wanted to say thank you so much again. I learned a lot. So
thank you for having me. I really enjoyed it.
Yeah, no, I really enjoyed it. I think everyone will have. And yeah, so as I said before, if it's okay with you, we'll upload this to the YouTube channel as well. So other people who couldn't make it can watch it. And we'll also advertise that you're looking for PhD students on the STEM Village Twitter page. And yeah, thank you again, and hopefully speak to you soon.
Great. Yeah. Thank you very much. And
Thank you.
enjoy the rest of the evening.
Thank you very much.
Bye.
And thanks everyone for attending. Bye, everybody.